Hello friends, welcome back to my channel Plant Pathology Pathshala. So in this video we will discuss about pattern triggered immunity. So as you all know that this is the first layer of plant immunity. So whenever a pathogen come in contact with or it attack the plant system, the first layer of immunity against a pathogen develop is the pattern triggered immunity or PTI, the first layer of immunity. Then how it is triggered? So it is triggered by recognition of PAMP or MAM molecule by the pattern recognition receptor present on the plant. So we have already discussed this, what is PAMP, what is MAMs. PAM means pathogen associated molecular patterns. So, and this MAMs are microbe associated molecular patterns. Both this PAM and MAM are same only. See, the common thing is every microbe means every pathogen is a microbe only. A fungi is a microorganism only, a virus is a microorganism, but all microorganisms are not pathogen. What is pathogen? Pathogen is that is able to cause disease in plant. Because it is causing some harmful effect, we are calling that pathogen. If it is not at all any doing any harmful effect, or it is not causing any harm, any disease, like the rhizobium is also a microorganism, it's a bacteria, but it is not causing disease in plant, but it helps in the symbiosis, this nitrogen fixation. It is a beneficial microorganism, it is not a pathogen. So all the PAMs are not MAPs. Okay. So this PAMs or MAMs are generally some signature motifs. Means PAMs और MAMs एक ऐसा मॉलिक्यूल है जो पैथोजन में पाया जाता है, जिससे जो हमारा प्लांट है रिकॉग्नाइज कर सकता है, एक सिग्नेचर चीज़ है. जैसे अगर हम बोलेंगे कि किसी इंसान का सिग्नेचर क्या है, उसका राउंड फेस है, उसके दो आइज़ हैं, कोई भी कैरेक्टरिस्टिक फीचर हो, वैसे ही पैम्प एक माइक्रो ऑर्गेनिज्म और एक पैथोजन का एक सिग्नेचर मॉलिक्यूल है. लाइक फॉर एग्जांपल फ्लैजेलिन present in the flagella, which is in the fungal flagella. Okay, so that is a signature molecule for the fungi. Peptidoglycan, where it is present? Peptidoglycan is specific for the bacterial cell wall. Okay, so if a peptidoglycan molecule is there, means definitely it is a bacteria only. So like that, the plant recognize the pathogen, and this recognition, how it recognizes, means plant also itself needs something that will recognize the PAMP or MAMP. So that recognition, that receptor is known as pattern recognition receptor, because it recognizes the signature pattern of the pathogen. That's why it is known as pattern recognition receptor. And these are present mostly on the membrane, on the outer membrane of the plant. Okay. So when a pathogen come in contact with the plant, the PAM molecule or MAM molecule are recognized by the plant receptor, that is the PRR, and in turn it will activate the first layer of plant immunity, that is plant innate immunity, that is the pattern triggered immunity or PTI. And its characteristic is it is a broad spectrum resistance, non-specific resistance against the pathogen. Broad spectrum means see, whenever any peptidoglycan molecule are there, when it is recognized by our PRR molecule, so it will activate a generalized defense against most of the bacterial pathogen. So this is a broad spectrum and non-specific resistance. Okay. So this is brief about the PTI. Next, So this is how the PTI signal is activated. First, the pathogen will attack. Okay, it will invade the plant. When the pathogen attack, it will release the MAMPs or PAMPs molecule, the signature conserved molecules, which is recognized by our pattern recognition receptor present on the cell surface. Okay, then this is a ligand, receptor ligand conformation. This protein interaction, protein receptor ligand or protein interaction will be there, and ultimately our PTI, the pattern trigger immunity, will be activated. Here some examples are given, like the chitin, flagellin, liposaccharide. These are the PAM molecules. Chitin is specific for fungi. Flagellin for fungi also in the bacteria. Then lipopolysaccharide. This is for the gram-negative bacteria. These are examples of PAMs. Then the major PRR involved. See, this for the chitin, the receptor present in the plant is CERK1, chitin receptor. Then this is the this flagellin. For flagellin, the PAMP is FLG22. Remember this, which is recognized specifically by the PRR FLS2. These are some examples. Okay. Then EFR, for EFR, this is EFTU receptors are there. So when this receptors are recognizing the particular PAM molecule, ultimately the ligand receptor complex will be activated, and finally the defense system is activated. The defense mechanism, that is the PTI, is activated. So after the activation, means after the ligand receptor complex formation, what are the signaling events that occur? See, for just like normal thing, like whenever anything happened, it need to be disseminated to other areas so that some action will be done. So like that, signals are generated in the plant cell. Then how the signals are, signaling events are there? So that signaling events are calcium influx, then ROS burst. These are the signaling event, early signaling event. Okay, that, and these signals are transmitted by some cascade, some pathways. Example of pathways like MAPK, CMAP, CAM pathway, then MAPK. These are the signaling pathway. We will discuss in detail how this means just like जब एक पैथोजन अटैक करता है तो उस सिग्नल को कि हाँ एक पैथोजन ने अटैक किया, नाउ जो हमारा रिसेप्टर और हमारा पैम मॉलिक्यूल इंटरेक्शन जो हुआ है, उसका जो सिग्नल है वो ट्रांसफ़र कैसे होगा डिफेंस जीन्स तक, वो जो पाथवे है वो सिग्नलिंग पाथवे के थ्रू वो ट्रांसफ़र हुआ, तभी जाकर डिफेंस जीन एक्टिवेट होगा. तो जो बीच वाला वो ब्रिजिंग मैकेनिज्म है, ये सिग्नलिंग जो कैस्केड्स है, ये सब वही सिग्नल्स ट्रांसफ़र करते हैं, लाइक मैप के that CAM pathway, G protein mediated signaling. So we will discuss all this pathway in detail. So जब क्या होता है, जब कैल्शियम इन्फ्लक्स मतलब कैल्शियम डिपोजिशन होता है सेल के अंदर, जब कोई पैथोजन अटैक होता है, सेल अपने कैल्शियम डिपोजिशन, कैल्शियम को इनटेक करना स्टार्ट कर देता है. कैल्शियम के अंदर जब इनटेक ज़्यादा हो जाता है तो कैल्शियम डिपेंडेंट प्रोटीन काइनेजेस एक्टिवेट हो जाता है, जो कि फर्दर डिफेंस प्रोटीन्स को, डिफेंस जीन्स को एक्टिवेट करता है. वैसे ही ROS बर्स्ट होने से क्या होगा, पैथोजन का ग्रोथ रेस्ट्रिक्ट करता है. अदर जीन्स एक्टिवेट करने के लिए ये मैप के कैस्केड, ये सब कैस्केड्स हेल्प करते हैं. ठीक है. देन अब ये कैस्केड एक्टिवेट होगा तो कैस्केड से क्या होगा, सिग्नल ट्रांसफ़र होगा. सिग्नल ट्रांसफ़र होकर वो सिग्नल पहुँचेगा कहाँ पे, डिफेंस जीन तक. So after when the person this defense, after this the defense gene expression will be there. Okay. So generally this PR protein expression, the phytoalexin biosynthesis will be there. Okay. These are the things that happen after perception or after receptor ligand complex formation. Okay. Next, what are the defense responses that actually happen after activation of PTI. So major outcomes are cell wall reinforcement. Cell wall reinforcement कैसे होता है? Like due to by Callose deposition. Callose are beta-1,3-glucan molecule that deposit over the cell wall and it will strengthen the cell wall. So this Callose deposition will occur and lignin deposition. So these are deposited over the cell wall to make it more strengthened. That will restrict the pathogen entry. Okay, at the first as a first line of defense. Then इससे क्या होगा? Ultimately pathogen growth will be restricted. See, the PTI is just like a security gate, just like a border only. At the border level, whatever normal security first line of defense the plant is imposing on the pathogen attack. Okay, so major things you have to remember. What are the defense activated? Generally the cell wall reinforcement. Okay, then that include the Callose deposition and lignin deposition. Next, about characteristics of the PTI. So PTI, as we have already discussed, this is the first line of defense that protect plant from wide range of pathogen. This is a broad spectrum defense mechanism. Okay, it's the first line of defense activated immediately after pathogen recognition. Then it is broad spectrum, non-specific. See, PAM molecules are conserved molecules for the plant. It's not like the effector or the specific toxins. So these PAM molecules are very general conserved signature molecules. So the defense activity is also very general, broad spectrum, non-specific one. Okay, then it induced defense mechanism that includes the production of PR protein and the phytoalexins, cell wall reinforcement. Okay, then it activates systemic immunity also. Then it is pattern based recognition because it is recognizing the PAM molecule. Okay, then significance same only, provide broad spectrum and non-specific resistance. It restrict the pathogen growth at early stage of infection. See, the ETI restrict the pathogen at advanced level, but at initial stage the PTI is restricting the pathogen. So it forms the primary defense barrier before activation of ETI. Okay, then this is the complete overview. When pathogen attack, it will secrete some PAMP molecule that may be the chitin, flagellin or LPS lipopolysaccharide that is recognized by our PRR molecule. The pattern recognition receptors present on the membrane. After recognition, it will form the receptor ligand complex that will generate the early signaling event. Early signaling event includes the calcium influx, ROS burst, then MAPK cascade or CAMP G protein mediated cascade, or the signaling cascade will be activated that will further activate the defense gene expression like PR protein and phytoalexin. Ultimately, the cell wall reinforcement will be occur that will restrict the pathogen growth, and finally enhanced plant survival will be there. So disease will be restricted. So this is all about the PTI, the pattern triggered immunity, the basal level of immunity or the first line of defense. This is the basic level of defense. Okay, the ETI is the advanced level of defense because it is due to the specific interaction between the effector and the R gene. Okay, we will discuss in detail about this in a separate video. Okay, in the next video only we will discuss. So I think now PTI is clear to you. So thank you for watching this video.